I'm here with Chip Slabow from Gator Bio. He's going to be showing us about the Gator label-free analysis system. This is very similar to our Octet QKE instrument that we are looking to replace in the next 5 years. So please, if you're interested in this instrument, please reach out to me by email. My email address is, or you can just call over to the UF ICBR and some. We're going, if you pre-registered, there's a lunch for you here in the Cancer and Genetics Research Complex, room 451, and it's Panera, so please come on over. I'm gonna let Chip get started. Thank you.
Thank you, Dr. Sampson. Hi, it's nice to meet you guys. And this is the Gator right in front of you. I'm going to do a little bit of a demo on here and go over. Oh, come on. So the agenda today, I'm going to go over Gator Bio history, over our biolayer technology, do a product overview, go into some applications, show you the advantages of Gator Bio and how we are bringing on next generation BLI, and go through a couple of Gator publications that we have. So this is Dr. Hawton and Bob Zuck. This is our CEO and our CTO. These are the guys that originally invented the biolayer interferometry. Our headquarters are in Palo Alto, California. ET Healthcare is our sister company. And Bob and Hong got into BLI to bring on the next generation. Their mission was to empower researchers by creating a more powerful, productive, and cost-effective BLI system. So in 2001, Hong Ken and Bob Zuck, they founded Forte Bio, and they started working on BLI prototypes. In 2006, Forte Bio introduced the Octet, and then Bob and Hong went into the IVD market. And they started creating IVD diagnostic instruments. And then in 2018, they founded Gator Bio, and they started working on next generation BLI. And I'm going to go over what next generation BLI is. And at the end of last year, we launched our second instrument called the Gator Plus. So real quick to talk about the IVD stuff that Bob and Hong are doing. This is a pylon. So this is a fluidics-free instrument, much like the Gator. This is a near patient diagnostic tool. And it actually uses the same probe technology that's in this instrument right here. So the way this works is we have a kit for certain biomarkers. We right now have a big cardiac panel. So it works, it's fully automated. You put in the patient sample, and the probe goes through the wells, and then it's read in an instrument. Super quick. In about 30 minutes, you can go through 60 samples. And this is what they were doing in between Forte Bio and Gator Bio. So right now, if you go to Google Scholar and type in biolayer interferometry, you'll see over 8,000 publications. And every single day, that number is growing. So BLI works using this principle, the reflectometric interference spectroscopy. So basically, let me get this going. So we have a probe, and light goes down the probe and is reflected off a couple different interfaces. So we have an optical base layer, and then it's reflected off the substrate between the probe and your buffer. And so what's detected is this interference pattern. So some of the wavelengths are constructively interfering, some are destructively interfering. And when interactions happen at this probe surface, that changes the interference pattern. It'll shift the interference pattern. So the detector is looking at this wave, and what is being displayed on the software is a change in the interference pattern. So this delta lambda versus time. So this is what a normal fensogram would look like. You have shift, so interference shift versus time. So our probe is 20 millimeters in length and 1 millimeter in diameter. And so we have our optical base layer here and the surface chemistry reflects off a couple of these different surfaces. When molecules bind to the probe, this is what we call the ligands. So when ligands are binding, that shifts the interference pattern to higher wavelengths. And what you see on the software, ligands are being loaded onto the probe, and you see this optical density changing versus time. Loaded, you can use that molecule to do other interactions. And so if an analyte, this molecule coming in here, the surface density, the optical density at the probe tip. And so you see another change in this interference pattern. So it's again shifting to longer wavelengths. This curve right here is what's called the association curve. Now, if you take this complex, put it into a buffer well, for example, that analyte should fall off the ligand. So if they're a very tight binder, it might take a very long time. But as this analyte falls off, you now see this decrease in the shift signal. And this is dissociation. So we have our association in blue and the dissociation curve is in red. So you're going to see this. I'm going to run a quick measurement in a second, but we're a plate-based technology, and the probes are picked up out of the green plate, which we call the max plate. And right now, the detector is running all 8 channels in parallel, and it's going dipping into from well to well, looking at the interference pattern changes. So the label-free analysis system that we offer comes with our software, instrumentation, and a robust list of consumables that I'll be getting into. So right now we have the Gator Prime and the Gator Plus. This right here is the Gator Prime. Both instruments use biolayer interferometry. And this is inside of here is temperature control. You can run assays at ambient temperature all the way up to 40 degrees C. Now, some of the differences between these two instruments. Our Gator Plus instrument can use 96 well and 384 well plates. So really where you see a big throughput difference is if you look at epitope binning. You can do a 12 by 12 on the 96 and a 17 by 17 on the Gator Plus. Now this 12 by 12 might be shocking for a second, but let me go back to here. The Gator allows you to put samples in both the 96 well plate by using the regeneration capability of these probes. You can use one column on your max plate for the probes. You would need 2 columns for the regeneration cycle. And then the other 80 wells in that max plate can be used for sample. So here's the dimensions of the instruments. As you can see here, this is our small guy, the Gator Prime. And we engineered this instrument so that you could use it in both orientations. So I have obviously this orientation happening here, but you can also turn the instrument on its side, and the depth here is 13 inches. So it really only takes 13 inches of bench space to run the Gator. So the reason why people are going to BLI right now is not only is it label free, which is very important, the instrument is very low maintenance. There's no fluidics. I always say this instrument is pretty much unbreakable. We've dropped one before. I've had customers do some really crazy things. You can really not, there's kind of no way to break this instrument. It's really, really strong, easy to use. Another cool thing about BLI is the sample is saved after analysis. So after the probe is dipped into the well and you get your centigrams, you can take that sample out and either preserve it or run it on other instruments. The type of sample that we can run is pretty much limitless. You can run supernatant, cell lysates, plasma, about as crude as you can think can be run on the Gator. We also do customized biosensors. So I'm going to show you a list of all the biosensors that we currently offer off the shelf, but we also do customized biosensors. And if anybody would like to talk to us about some ideas you have, we're a pretty small company, and so the R&D team we have is really, they're ambitious about creating biosensors to help people's research, whether it's some type of RBD for COVID, where we've been doing a lot of COVID biosensors. The software is very easy to operate. I'll show you that in a second. And the biosensors that we offer can be regenerated and reused many times. So right now, BLI is used in pretty much every single department you can think of. Here's just a list of departments that are using BLI. This list is constantly growing. People are learning what BLI is, and they're getting creative. But right now, this is our list. Here are the probes that we offer. So I have the dynamic range listed, regeneration capabilities, but we just released 2 new probes last month, the Flex assay and NAVX. And I'll go into those a little bit later. But back to the custom made. So the SARS RBD, we're doing anti-RBD on the probe. Some of our researchers need anti-rabbit, anti-rat, anti-fly. So we make these customized biosensors for these customers. So quickly, what is regeneration? When the probe is sent to you in a tray, it is pre-coated with the capture molecule. For this example, this probe is pre-coated with anti-human FC. So if you run an experiment, you might get some antibody, target antigen, some non-specific bindings. When you regenerate the probe, you strip all of those assay molecules and you go back to the pre-coated anti-human FC. Now with the streptavidin probes and the immune reactive probes, it's slightly different. So the probe is pre-coated with streptavidin. During your experiment, you'll have some bioaccumulated ligand that is loaded onto the streptavidin. Then you'll have your assay molecules like the antibodies, maybe some non-specific binding. When you run regeneration, you do not strip off the bioaccumulated antigen. So this is well known. BLI has been like this for a long time. Our Flex assay probes, which I'll have a slide in a little bit, there's a way to completely regenerate streptavidin and go back to just streptavidin on the probe tip. So the way concentration or titer is determined using BLI is through our quantitation assay. So this is a pre-built template. And if you wanted to run, this is an example of a full plate. So you may run 2 columns of standard, and then your other 80 wells may have unknowns in there. So this takes 5 seconds literally to build this plate in the software. In about 20 minutes, you can run through this full plate. And the way it works is it looks at initial binding rate of your standard. So you put your concentrations of your standards, and then this binding rate curve, the standard curve, is created by the software, and then your unknowns are plotted on that standard curve. So I have this scenario. One day I run into my lab manager. She wants me to generate 30 unknown antibody concentrations within 10 hours. What should I do? ELISA, HPLC, Western blotting. All those techniques take a very long time. With Gator BLI, you can set up this plate, run it, and do the analysis in just about 20 minutes. So the way Kinetics works on Gator is, I kind of showed this in the previous video. So you would have a loaded ligand. So in this example here, we have a bioaccumulated antigen. So that is loaded onto the probe. And then if we want to get some kinetic constants, we would run a series dilution of the analyte. So in this example, it's an antibody. So this series dilution has an association step and a dissociation step. And after this is run, we will get KI, KOF, and KD. So I actually have a really neat example that takes 2 seconds to set up right here. So here's our software. So I'm going to start a K assay. Is loading. So I'm going to actually do a biotinylated protein A and human IgG kinetics assay. So I'm going to do a concentration series starting at 25 micrograms per mil. It's going to be a twofold. This is my human IgG. And then here I have, I'm declaring my probes are in the max plate. So max plate, sample plate. Now for my assay steps, I'm going to do a quick 30-second baseline. Oops. Now my next step is going to be loading. So I'm going to load for 120 seconds. Then I'm going to do a quick 30-second wash back in that original buffer column. Now here will be my association. I'm going to do that for 120 seconds. And then, whoops, I need a dissociation. So column 4 will be dissociation. And now it's ready. I can go step by step and just make sure that I built my assay correctly. So I'm going, let's do, oh, sorry. Baseline, loading, wash, association, dissociation. So I set that up here. Normally you would not leave this open, but I'm going to leave it open so you can kind of see it move around. Okay, so let's go back. So in about 10 minutes, we can come back and check on what this is going to look like. Okay. So another pre-built assay in the Gator software is epitope binning. So you can run sandwich assays, tandem assays. This is a 4x4 sandwich, 4x4 tandem. So this is what your assay is going to look like. Now for the data analysis, this is an 8x8 example. So after the assay is run, you will learn everything you need about your epitopes. So the software does all your binning for you. You can get your epitope networks, look at binding rates, look at all the kinetics. Something I actually meant to say real quick. Something that is really, really helpful in some instances on the Gator BLI. When you run kinetics, you can actually quantitate any of your steps. So if you have a standard curve saved, I'm looking at kinetics here, but I can actually do quantitation on this step. So I can calculate the initial binding rates and get quantitation of any single step. So that's something pretty new about BLI. So I just have some quick, interesting examples of what people are using BLI for. So this is an example of looking at the Bacterial infection and looking at the protective, like how it protects against smallpox and how the epitopes of the antibodies made from the Bacterial infection also target the smallpox virus. And so what they did was they took the L1 residue on the Bacteria and they did mutagenesis, biotinylated, or eventually used the nickel and TA. They loaded their mutated L1 residues and then looked at their large database of different antibodies. And they bin their epitopes of the antibodies that they were creating. Another one here is looking at the affinity of 5-UTR RNA with the elongated factor 1A. So they biotinylated this RNA strand, loaded it onto streptavidin, and they looked at the binding of elongated factor 1A, and I think there's a couple other ones they did. They looked at other factors, but very quick, very easy. You can look at the binding of the RNA using the streptavidin probes. There are also ACR or anti-CRISPR candidates. So this is another interesting assay that people are working on. There's this protein that inhibits the Cas9 complex and its activity. And what they did was they looked at different proteins and looked at the way they inhibit the Cas9 in a DNA cleavage assay. And so here we have the response and the concentration of the ACRs. So if you're doing HPLC, Gator's going to be a really amazing tool for you. This is an example using Proteina. So this is a regeneration sensogram. So the same probe was used 50 times, looking at 300 micrograms per mil of human IgG. And you see this really, really strong correlation between every single run. So the regeneration capabilities are very great with the Proteina. And then here we see the dynamic range of Gator BLI is 4 logs using the Proteina, 1.9 logs HPLC. The lower limit is 0.02, much lower than we can get on HPLC. And the time you're saving to prep, run your assay, and do the analysis on Gator is less than 30 minutes. And if you're lucky, that's going to take 8 hours on HPLC. So we just released the NAVX Probe. This is something that we are very excited about. We can do tighter determinations covering 4+ logs, and you can run a whole plate in a matter of minutes. So you can regenerate these NAVX probes. It's binding to serotypes 1 through 10 minus 9. And if you look here on the CVs, you're less than 2% through most of this dynamic range. And when you're getting to the lower limit, we're about 10% CV. So this is what I was talking about before, reactivating your streptavidin. And this is our new Flex assay probe. So before, when you had a normal streptavidin probe, you do the regeneration and you still have your biotinylated liquid. There's no way to remove that bond. But what we created here is a fully regenerated streptavidin probe. So after you run your assay, you strip the streptavidin and reactivate and load streptavidin back onto the probe. So there's many different ways this is going to help researchers. So saving time, cost, assay development is much quicker and easier using this Flex assay platform. Another thing that we're doing is the mouse FC probes that we have we've innovated the mouse FC probes such that our dynamic range goes down to 0.02 micrograms per mil. Traditional BLI cannot get below 1. The regeneration capabilities as well. You cannot regenerate traditional BLI mouse FC probes, but without Gator mouse FC probes, you can regenerate. So we have small molecule probes. So if you're looking at interactions with small molecules, we have a streptavidin probe specifically for small molecules. And if you see here, this is just an example of loading the same sample of 52 kilodalton protein. So we're seeing a 15 nanometer response in the loading versus the first generation of BLI, we're looking at 4 nanometers. Here you see a very nice dose response using our SMAP probe, the small molecule probes. And this example here, this 332 Dalton molecule, cannot be seen. The signal to noise ratio is too large on the first generation of BLI. We also have a picomolar buffer, so if you have really tight binders, this is necessary to get these long dissociation steps. Sometimes with Gator, you can actually run the only limiting factor is the evaporation of your buffer. So what would you say, 2 hours you can run you can run a dissociation step 2 hours before you see any type of evaporation effect. And what this picomolar buffer does is it stabilizes the drift. So when you're looking at these very tight binders, 2 hours, you might just see a couple % difference in the dissociation. And so a stable baseline is very, very important for these type binders. So our proprietary picomolar buffer is what you're looking for when you have these very tight, small kb interactions. So what we're doing with next generation BLIs, it's a lot that has to do with what we're doing with the probes. So we increase the surface area by 3 times compared to the traditional BLI. We've also improved the optical layer that allows us to detect smaller refractive index changes. And what we're doing on the surface, we're changing the surface chemistry to allow for better signal to noise ratio, different types of capture molecules. And another thing we did is we created this V groove. So in the picker, so right now, these probes are up in the picker, right? And the optical fibers are shining through the probes. This V groove allows for perfect alignment every single time. So BLI is a pretty sensitive instrument. The physics makes so that this alignment is very important. And so we did this V groove allows for 3 points of contact, and your probes will be in the same position relative to your optics. So I kind of spoke about this before with the snap probes. We've improved signal to noise. So this is some of the publications right now that people are coming out with. This is Gator publications right here. I can send you this list. We put this together after our talk with Dr. Sampson. These are some really cool papers. I can also send you this list. So this is people using BLI with lipid nanoparticles and specifically cancer research. So with that, I'd like to say thank you. I think it's really cool to have a Gator come to the UF. And so thank you very much for coming, and go Gators. If you have any questions, I'd love to sit up here and talk. I will just show real quick what this assay looks like. I'm going to put this on the screen. The result. And I ran this assay before, so this is exactly what it looks like. And so I just want to show you how quick the analysis is. Association step. I'm going to blow through this, so if you're not following along, don't worry about it. So I have one reference probe. I'm going to subtract my reference. And then I'm going to do a global fit. We can talk more about this later. But so remember, I'm looking at the affinity between protein A and human IgG. And the KD I'm getting is 6.52 times 10 to the minus 6, which I think literature says 7. So I think we're spot on there today. So if you have any questions, I'd love to sit up and talk. You want to look at the analysis? Okay, cool. Okay, so we'll just use this example. I'm going to create a brand new analysis. Okay, so you want me just, as I'm going through, just talk a little bit more about these steps. So since we're looking at KD, really the only 2 important steps are association and dissociation, right? So one of the things I'm going to do here is I want to align all my signals to the start of association. So that's what this aligned y-axis does. So now if I look at the process, now we have our two important steps. The interstep connection or correction, that's typically if you have buffer changes and you have like a big difference, that will, because theoretically you need to have the end of association start or end with the start of dissociation. Those two signals should be the same. So the interstep correction does that. You can do some smoothing if you need to. The raw data correction, I can take data off. I could, for example, this is what's cool about Kinetics. So I can now, I just cut 300 seconds off my association and 300 seconds off my dissociation. But using the intercept correction, now it puts those, it stitches those two signals together. And so the reference, this signal right here is just buffer. So I loaded my biotinylated protein A, but it just ran through buffer. So I'm going to label this my reference probe. And now if I highlight all the probes, edit formula, selected probes minus my reference probe. So now these are ready to be fitted. So in the parameters, if you're looking at KD between two molecules, you want to have your concentration series be in the appropriate range. And then what you want to do is do a global fit. So the global fit is going to use all those centigrade, all those curves, and the algorithm is going to come out with one single KD. So if I do local fit, it fits each curve independently. So the full, because during K on, you have, since it's a dynamic interaction, you do have some K off happening. So the equation for K on has K off in it, but you can do the partial fit. So full is going to look at that interaction the way it happens in real life. Partial will look at just K on and fit that, and then look at just K off. So it fits them independently of each other. But for this example, I would want to do a global fit. So now, it changed by taking those 300 seconds off, it changed the KD by a few percentages. So that's pretty much it. So in the software, you get the K, all the errors, KD, R squared, a lot of parameters that you might not even care about. And then what I can do is I can create, so first I could just export the entire everything to Excel. But what I can also do, what's kind of cool in the software, is we have what we call our canvas. So I can create this canvas and I can get really artistic with it if I want. So let's see. So let's say, you have a big database of antibodies that you're trying to, do some K rankings. I can create a canvas, save this template, and then as soon as the Gator's done running, it will do the analysis the exact same way and then export this canvas. So this is a pretty neat tool. So that's pretty much it for the kinetics analysis. Do you have any more questions or want to go over anything else? Okay, cool. Anyone else? Well, I have my email on the front of this. So if anyone on Zoom or anyone here, let me just go. So this is my email here. Let me open up Zoom and see if there's, all right, nothing in the chat. think that it's definitely a few things. The optical layer, the new optical layer that was created, that along with the surface area increase. Yeah. So for the 96 well, 180 to 220 microliters. For 384, I don't have to pull the plate out. 384, you can actually go all the way down to 40 microliters. No, that's just, you can have different capture molecules. Yeah. Mhm.
And the plates, are they purchased or are they purchased?
Yeah, so these are grinder plates. I recommend the polypropylene 96 well, flat bottom. These plates we make. So this is what we call the max plate. And yeah, you can put sample in here, and just the way I just set up an assay, I can put my samples, do whatever in the max plate, so I can utilize both the grinder plate and then the Gator Bio plate. Yes, so yeah, there's a protective sucrose layer, and it takes about 5 minutes. We recommend a minimum of 5 minutes in buffer. So whatever assay buffer you're using, pretty much before I run any BLI, I figure out what probes I want to set this up, and these probes can sit in buffer all day. So yeah, so you want to get that going. It's probably, it would depend on what type of assay you're doing. I have customers that absolutely do that.
Just keep the sensors
In buffer, yeah. Yeah, so I don't know where the lid is. Yeah, basically, the tray comes filled with sensors with a lid. So you just keep them in buffer, put a lid on, put them in the fridge. But yeah, you would have to test if that's going to work for your assay.
And so when you're doing the step down regeneration,
Mhm.
that's just a special product.
Yeah, so it's a kit, and there's I think 5 columns of different types of buffers, and you're basically stripping the streptavidin and then reloading streptavidin. So this is a very new probe. I actually have not even used the kit yet. I think we're saying, again, this is probably dependent on the type of assay that you're doing. I think are we saying 30? At least 10? Yeah, this is really cool. Yeah. Yeah, so you can have your tray, ready to go with your streptavidin kit, and just keep reusing it. Put a lid on it, put it in the fridge. Yep.
I think that's one of the most expensive parts of these.
Absolutely. Yeah. And yeah, I think for the flex assay, I've heard from customers they can now do so much more optimization before they get into their assay. Because before they were like, oh, how many sensors do I want to go through? Because if I'm bored all day, I could check 10 different buffers for the, for my kinetics. I can try all these different concentrations. If you're worried about going through probes, and the strep probes are actually a little bit more expensive than the other probes we have. This might cost you $1,000 a day if you're going through a big list of agents you have. With the Flex assay, I mean, I don't know what the table on here, but like this cost you're saving so much money. And just the freedom of being able to go through different antigens and not worry about wasting probes. Well, I have an email on here. Yeah, anyone can reach out anytime. I can send you over some product notes.
You mentioned to people online about
Yeah, thanks for reminding me. So the instrument is going to be here on campus. I'll be here for the next 2 days. The instrument will be in Dr. Sampson's lab. And send me an email, send Dr. Sampson an email. We can run some of your samples. You can come check out the instrument in person. And yeah, we'll just go from there. Monoclonal antibody core. That's where Dr. Sampson's lab is. All right. Well, I think if there's no more questions. Thanks. Oh, go ahead. Yeah, so when yeah, so I run the biosensor Proteina and IgG. I run that all the time. So for one, I have a lot of that data, so I know very well the kinetics of those two molecules. But in your case, so a brand new Gator goes through QC. There's a lot of different assays that are run on it, and you get copies of what that data is supposed to look like. And so at any point, you can always run those samples and see if your instrument is still behaving the same way it did on day one. If that answers your question. We're working on it. We will have that. Okay. At this point, I would just come out with that.
So tell me if you license with your like capture license. Do you sell a license?
We don't sell a license necessarily per se, but we would easily work with you to develop some type of assay that would mimic what you're seeing on the license. It's definitely, yeah, it's cheaper. It's going to take way less time. Once you set up your plate, you just press go, and there's no more human interaction.
caution.
Yeah.
We just did a study with a major pharmaceutical company. Comparison of the same kind of drugs. And it's awesome.
So is it possible, do you have to test different concentrations of your antibody, loading it to the sensor to make sure you haven't ever overloaded it or underloaded it?
Yeah, so typically, if I come across two molecules that I haven't run BLI on yet, I will definitely do a loading optimization assay first. With the antibodies, I pretty much run 3 different concentrations, maybe 10 micrograms per mil, 2.5, and then maybe 1. And then during that assay, what I want to see is I want to see what the final response is. So what a saturated probe, what immunogen shift that has. And then in literature, you'll see the number 60%, especially with antibodies, you don't want to crowd the surface, the probe surface. So you would want to take your final response, take 60% of that, and you also don't want to load too fast. So I typically tell my customers 120 seconds is a very good loading time. I'll have customers send me data where their loading time is like 10 seconds because the concentration is just too high. So they're hitting that 60% mark, but they're loading too fast, and that's not a reliable way to load, especially antibodies. So yeah, I would run that loading assay, and then I would get a rough idea of KD. So then once I know the loading concentration, I'd run another high, medium, low concentration with analog. Do local fits on those 3 concentrations, and then I'll give me a pretty good idea of what KD is, at least like in the ballpark. And then I make my concentration series span that KD. Oh yeah. Yep. Yeah, you can pretty much about as crude as you can think. I mean, of course, you're gonna have to have the appropriate references if you have, some non-specific binding to either the probe surface, capture molecule. But what's cool with BLI, so you have these 8 channels, you can set up multiple controls, and then in the software, I can subtract all those signals from my final analyte signal.
When you're loading, trying to quantify different antibodies, do you have a sensor that can measure?
We're not doing IgM, are we? That could be a custom sensor, right?
Going back on our list right now, proven A, proven T, also we are expecting out. That we know about, but we do need
All right. Well, thanks again. Hope you guys are hungry. And yeah, I'll be around for 2 more days. Thank you. Thank you guys. I'm getting off here.